So the infrastructure project basically has two goals. One is what drugs might be useful for Angelman syndrome, and how would we find more drugs for Angelman syndrome? So we've talked a lot about different kinds of molecular therapies, and especially yesterday at the scientific symposium, where we hear a lot about molecular therapies. And molecular therapies are a little bit different than drugs. They work on the level of these molecules. They work with the DNA and the RNA. And certainly there's been a lot of active area with these kinds of things. And so a while ago, as Allison had already said, we published a paper with artificial transcription factors. There are people working on gene therapy for Angelman, and we've heard a lot about those kinds of approaches. Then, of course, things like antisense oligonucleotides have been developed to chop off the end of that antisense transcript and reactivate UBE3A, and that's in clinical trial. A fantastic achievement in the history of this disorder. And some things that we're working in our lab on are things like using CRISPR proteins. In our lab, we're not actually using the CRISPR-Cas9 for gene editing, but we're using a different kind of CRISPR that is Cas13D, and that's also doing the job of cutting off that antisense transcript. And of course, there are other groups that are also working with CRISPR proteins as well, usually trying to chop something on the DNA or using some other kind of vectorized system to inhibit the stopper, that antisense transcript, and allow UBE3A to be expressed in the brain. So those are all molecular therapies, and they hold a lot of promise for the future of this disorder. And so we and many others are trying to work very hard to develop these kinds of therapies and test their efficacy. But all of these therapies have one thing in common, and that is they're trying to reactivate UBE3A in the brain. And so we need to see how successful that's going to be for everybody. But we might want to think about other things aside from just straight reactivation of UBE3A. So certainly, we hope that UBE3A reactivation is going to affect all the children and bring about a great cure. But there may be some domains that it doesn't work as well in as others, and so we might need to supplement that. And there also may be drugs out there right now that could be beneficial to our community, and we would like to know that if we could access those too. So there are still a lot of reasons to think about drug therapy. And so in thinking about what kinds of drugs would we need, maybe another question that we could ask is, what does UBE3A do? And are those kinds of functions things that are druggable? And so in a paper that we published just a couple of years ago, we looked at the genes that are expressed in the brain that are different in Angelman syndrome versus neurotypical. And in our study, we came up with these different kinds of functions for what the loss of UBE3A was doing. So we know that UBE3A is a ubiquitin ligase, and it's supposed to interact with proteins, yeah. But what does that all do? And so in yellow, we see some different kinds of functional domains. And one is the proteasome, and we know that that's involved in degrading proteins. But we also see that changes in UBE3A change things like transcription and translation, or they affect the synapse, or they affect the mTOR pathway, which affects metabolism and cell maintenance. So these kinds of things or processes that are altered in Angelman syndrome. And so could there be drugs that could affect these processes so that even if reinstating, say, UBE3A in the brain at this stage doesn't help as much as we had hoped, maybe we could affect one of these processes. Or maybe, like I say, there's just other drugs out there that could augment what we're trying to do with UBE3A therapy. So this is a question, is can we treat these so-called downstream processes? And so aside from this kind of fancy gene therapy, gene editing, epigenetic editing, and antisense oligonucleotides, these are all therapies that are very good at aiming at very specific kinds of genes and trying to either fix those genes or change their expression at the DNA level. And so maybe a good therapy when you're really sure that you know what the gene is that you're going after, like we're trying to go after UBE3A. But if we're trying to affect these other processes, there might be other genes involved. And we might not know exactly what they are today. So we might not be able to target exactly that gene today with a molecular therapy, but we could use small molecule drug therapy. We meaning the community, right? But where do we find these drugs that that could be useful for Angelman syndrome? So, one example of where drugs come from for Angelman syndrome are companies like Ovid. And, they're certainly not the only one, but we could use Ovid as a really good example. So Ovid had a drug. They knew what their drug does, right? They knew the molecular biology of that and the pharmaceutical chemistry part of it. And they said, we think we have a drug that could be useful for Angelman syndrome. And so they bring that to FAST, they bring that to the community, and they try to push that forward. And most critically in this story, Ovid was able to take this drug into clinical trial. And so let's say one of the pathways is there's companies out there, they're working with some drug, they think that it could have some efficacy for Angelman syndrome. Now, they brought it through clinical trial. And so, we can look at the outcomes of those clinical trials, but we know that, as Allison just nicely said, there's a lot of drugs that are in this earlier stage, kind of in the drug discovery stage or the preclinical stage. And not all of those drugs are supported to go through clinical trial, and there's a lot of them. So one of the things that the infrastructure program really focuses on is how do we take these drugs where there's not a company that is able to take it through a clinical trial, and we want to know, is this going to be a drug that's going to be useful to go into clinical trial, right? Because clinical trials are expensive. So that's really the space that the infrastructure program is operating in, is in this preclinical kind of drug discovery stage where maybe a company has a drug and says, we think this will be useful for Angelman syndrome. They might have some preliminary data that says this is actually useful for Angelman syndrome. And so what we do, and when I say we, it's mostly Jill Silverman's lab that does this, because she's a real treasure for us in this community in what she can do with these drugs and her translational assays. And so we can take these drugs and we can analyze them all in the same lab with the same set of assays so that when we say, this drug is working, and we can compare it using the same kind of assays, the mouse, the rat, whatever we do with it, it's always going to be the same. So we treat all the drugs the same. So instead of saying this lab says it works, this lab says it doesn't work, we're just going to look at it in the lab at UC Davis. And that way we can compare these different kinds of drugs and see which ones are efficacious in our hands. So with that, we're going to play this. We're going to play the video, I think, right now. So this is what happens in Jill Silverman's lab when they get drugs.
Hi everyone, I am Jill Silverman. I'm really sorry to have missed this meeting in person. I broke a limb. I look forward to it a lot and being with all of you each year and sharing on this time together. But I'm really happy to see that everybody got there safe and that it's still working out. And I'm also really excited and short on time to talk to you about this infrastructure award that FAST generously awarded to Dr. Siegel, Dr. Fink, and I to test a variety of different compounds, a variety of different potential mechanisms of pharmaceutical treatment for Angelman syndrome. We are proposing to test them in vitro in cells and in vivo in rats and mice. And this is all going to exponentially improve the number of clinical trials that we have going and the number of therapeutics that we have testing. And it's really a huge endeavor. And so I'm going to talk really fast about about it. And so you know what our lab does translational outcome measures, meaning we test the various endpoints that will prove whether a drug is efficacious or whether it's safe and is actually going to do some improvement in either a curative way or maybe just a symptom way where we're improving, just seizures or just learning and memory. But in Angelman syndrome, there is no just. And so any improvements are going to be improvements in the quality of life of you and your loved one. These are not small measures. These are not small asks. We know that. And thus, as it takes a village for you to take care of your families, it takes us a village of scientists and resources to make these discoveries. You heard from a lot of these people yesterday, Dr. Berg and her work with the rat model, and Dr. Copping and all of her work in using EEG as a translational biomarker, Dr. Adhikari and her work with Joe Anderson and hematopoietic stem cells. And you heard from Tim Fenton, who is soon to becoming a doctor or doctoral candidate. And you heard his work with lovastatin and some other of his in vitro outcomes that he's discovered, or cellular outcomes. And so that was all done through the infrastructure grant. And so here on the bottom of this slide is a snapshot of cells and of how we measure learning and memory deficits and motor deficits and seizures. And so I have very little time to talk to you about a lot of ways that we tell whether a drug is safe and working and useful and purposeful. So let's get right to it. So Jessica Duess, you heard from Jessica yesterday, talk about motor outcomes. She's really been my motor soulmate as throughout my over 20-year career studying behavioral neuroscience. We studied a lot of complex behaviors of various types of learning and memory and various types of social interaction, attention, all kinds of things that are very, require sensory motor systems to understand, take in some information, have your brain process it, and then put out a different behavior yourself with sensory motor processes. And so I've been studying that my whole life, and I've also been really obsessed or sort of held on to the fact that motor influences almost every single part of this pathway. And so Jessica was also very interested in how different aspects of motor, fine motor, gross motor, hypotonia, ataxia, and how that is affecting Angelman syndrome kiddos and their ambulation as they develop, and then also how they may become more sedentary and actually lose ambulation as they get older. And so we sat, and I found this fascinating. So we had a lot of interactive calls where we went through her human outcomes and various outcomes that I could take in the mouse and rats, and sort of designed what would be the most optimal outcomes that we could get for motor assays. And so a lot of them we are using now. They're not all here on the slide, but I just wanted to give a shout out to all of that. One that we are using that is a big part of the infrastructure grant, and that you heard Tim Fenton talk about yesterday with lovastatin, is we're using the Digigate, and we're using stride and stride length and frequency and cadence and braking and all of these aspects that come out of the Digigate. We're able to use that to test if therapeutics work. We also published a paper just recently with Dr. Duess and my former PhD student, Dr. Petkova, that actually showed how Angelman syndrome mice and rats have increased stride length and decreased stride frequency. And so another really important component that is interesting about the rats that had not been previously published was that one of the first discoveries in Angelman syndrome when we first created a mouse, and as Allison says, it was a great decade to be an Angelman syndrome mouse when she found out when she had Quincy, because at that time they had discovered LTP, so they knew the underlying physiology of learning and memory deficit. They found that deficit in the mouse model, and then they were able to cure it a lot. Well, so for the rat model, we wanted to make sure that same electrophysiological deficit, or that same underlying mechanism of learning and memory, was there in the rat. And so this year we published and showed that it does indeed exist in the hippocampus, long-term potentiation deficits. But the more exciting part, so the actual behavior where we see a learning and memory deficit, took place on this right side. Here you see this rat performing the iPad task. In this task, you have to ask it which image is correct, and by correct, they get a strawberry milkshake reward. The plane is correct here, so they're actually pressing the plane all the time. The Angelman syndrome rat model took, I mean, they eventually got it, but they took a really long time to get it, 2.5 times as long, and they made a lot more errors on the way. And so we discovered that to be hugely important in the rat model, and strong and visible. And so we're able to get at that as an outcome measure. Nicole Copping talked to you a lot about EEG yesterday. She's using EEG as a biomarker. This was discovered by Ann Anderson, and Michael Sidorov had discovered it in the animal model. Shefali Giste has also shown it. And so we have a lot of different collaborators that have shown this to be rigorous and reliable. We're really excited that we can see it in the mice and rats. What Nicole does that's really interesting and makes it a little more translational is that she's not using an electrode that's implanted deep in the brain to look at a field potential. She's using a screw right on the skull and implanting the animals with this little barcode here, so that it's sort of just like the supermarket, and then they live in their home cage. They're not tethered to anything, so they live in their home cage, and then they're able to do various behavioral tasks like auditory potentials, like you heard about from Sasha Keys, or visual potentials. And so the EEG paired with behavior is a really exciting way to go. It's an exciting outcome measure for Angelman syndrome. The elevated delta power that I talked to you about from Ann Anderson and Mike Sidorov, Nicole also used this method and was able to see it reproduced here, where we saw the elevated delta in the UB3A mouse. So that's a third lab that's reproducing it. So we have a lot of confidence that is, if we have a lot of confidence, I mean, we can say with some confirmation that it's a phenotype. We actually also saw this be reversed by Dr. Anderson's hematopoietic stem cell rescue. So we're very excited about how delta power is going to inform us of pharmaceutical treatment going forward. Okay, I'm right at 1 minute over. I'm sorry, Dave. So the last phenotype is sleep. Dr. Copping discovered that sleep is so important, right? Your kids have sleep problems, and you have sleep problems, and it's terrible. We all have sleep problems. But sleep is important for cognition. It's important for your kids. It's important for all of us to be sane. And therefore, these sleep deficits that have been observed in Angelman syndrome are something we really want to target. Even if we can just fix sleep, it would be a huge change for all of you and all of us. And so what we know for Angelman syndrome is that it takes longer for those kiddos to go to sleep. When it takes longer for them to go to sleep, they also spend less time in that deep stage of sleep where their learning and memory is condensed, and learning and memory takes place. And we want them to take more time, want them to be more time in it, not less. So it takes them longer to get to sleep. They spend less time in the important stage. And they also sleep on total less. And those are all things that we can change. There is a particular sleep wave that is found during sleep stages that is hypothesized to be indicative of when learning and memory occurs. These are called sleep spindles. This year, Nicole Popping reported that she discovered reduced sleep spindles in the AS mouse model. Really excited to find compounds that will change or reverse and rescue that. And so with that, I'm going to wrap it up and allow Dave to talk to you about all the exciting compounds that we are testing and answer any questions. Thanks.
Okay, thanks to Jill. We're really fortunate at UC Davis to have Jill Silverman, who is a real expert in this area and has a real focus on translational assays to understand what the drugs and what the therapies are doing, because she has a very strong bias that we don't want to just learn if it makes the mice better. We want to have predictions that say that this is going to work in people. And that's really been the major focus of her neuroscience research. And even though we don't have Jill with us here up on stage today, we have three members of her lab here. So we have Anna Kari, Tim Fenton, and Liz Berg. Could you guys please stand up? So these are the people that are actually doing these assays in the lab and are actually trying to help to understand what these drugs and what these therapies are doing and if they're going to be useful in Angelman syndrome. And so I'm just going to show this one slide of a lot of different assays from work that was done by Tim Fenton. And with all these assays that Jill just described, we could look at drugs like Paxilene or look at a drug like LB100 and lovastatin. These all came from companies that thought that these drugs would be useful for Angelman syndrome, and we can analyze them using this infrastructure process and find drugs like lovastatin that seem like they might be useful in some of those domains, and so we want to follow up on that further. So some of the drugs come in that way. They come in from a company that has a drug that they think might be useful, but how would we find other drugs if it doesn't come from a company where they think, hey, my drug might be useful? How could we find other kinds of drugs that might be useful in the treatment of Angelman syndrome? So our project works on that as well. So famously, Ben Philpott published this drug screen that he did, where he basically took a library of different kinds of drugs. I think there were 2,500 drugs in this library. And you could see that some of them were off-patent drugs or CNS drugs or different kinds of drugs that already were in existence. Okay, and he did this drug screen of these 2,500 compounds, and he found one drug that could actually turn on UB3A, and that eventually became the topotecan drug that you've no doubt heard a lot about. And so this is an example of a drug screen where we can screen for some kind of feature and say, does this drug do this effect? Now, in this case, again, the feature that he screened for was, could we turn on UBE3A? So like those molecular therapies that I was talking about, and you're going to hear more about, this was a screen to reactivate UBE3A. But like I said, are there other things that we should be looking at now that are not just UBE3A? So how do we go about thinking about that? So we can, so for these kinds of assays, we can't test 2,500 compounds in the mice right off the bat, although that would be the best way to do it, but that's a lot of mice and a lot of time. So we don't have that kind of time. So for these, we use cell assays. And specifically, we want to use neurons. So we can take a drug library, and we could screen 2,500 or some number of compounds on neuronal cells in a dish. But the question is, what kinds of assays would we use to know if that was working? So if we have to turn on UB3A, that's one kind of assay. But let's say we don't want to, that's not the assay. So what other kinds of assays could we use at the cell level, right? We don't know if the cell can help that person to talk. So what can we actually look at that can tell us that something's getting better? And so these are called cell autonomous assays, where basically, you just need that cell, and it tells you something is going to help. And so one of the things that we could look at in that left panel is neuronal morphology. And this is something called the Scholl assay. And basically, if you look at that kind of spidery picture, that's supposed to represent a neuron. And the neurons have these branches that come out so that they contact the other neurons. And then sometimes those processes branch off into different directions. And this is an analysis where we can look at the number of branches that they have. We could look at how long they are. We could look, are they close to the cell body? Are they further away? And so this is the analysis that's looking at this kind of neuronal morphology. And so this is one of the kinds of assays that we're going to use to look at the cells to see if, when we treat them with these drugs, do we see a difference between neurotypical neurons and Angelman neurons, and can we find drugs that make the Angelman neurons more like neurotypical neurons? And then on the other side, oxidative stress. So this is just another assay. It's not only these two assays, but these are examples. Okay, so this was some work by Eric Klan's group and collaborators. They were looking at so-called oxidative stress in these cells. And basically, oxidative stress is something that all of our cells do, but then the cell kind of works with this oxidative stress and tries to reduce it. Without going into the details of the assay, you can see there's a line at the top that says wild type, and there's a line on the third line that says Angelman syndrome. And using this assay, they were able to induce this oxidative stress, and they could look at it with a certain kind of dye. And basically, you could see that the Angelman syndrome neurons were different than the wild type neurons. And so now this is an assay where we see a difference, and we can put drugs on these cells and say which of these drugs can rescue this oxidative stress phenotype. And so these are the kinds of assays that we're going to be using with cells that are in culture to start screening libraries for new drugs that we didn't even know about might be useful. We could screen libraries of FDA-approved drugs so that if we find drugs that are useful and already FDA-approved, and they look like they're working in this cell-based assay, we could fast-track those to now go into the kinds of assays that Dr. Silverman has just described and then test them in the mice. And then those would be high-priority candidates to then go into clinical trial. So this is how we're trying to accelerate all of this drug development. And again, these are the kinds of areas that we want to try to target with the drugs with these different kinds of assays. And so in the last slide here, I just want to show that another kind of cell autonomous assay that we can run is looking at different kinds of biomarkers. And so we know that there's some proteins that have been characterized in the literature over the past several years of the community doing research on Angelman syndrome. We know that there's some proteins that have altered expression patterns. And so this is another kind of thing that we could ask the cells. If we put these drugs onto the cells, do they have an expression pattern now that is more like a neurotypical neuron and less like an Angelman syndrome neuron? So between those kinds of physiological morphology and oxidation, and let's say these kinds of different processes, we can also look on the level of the molecules and ask, is this drug treatment actually bringing the cell closer to something that looks like a neurotypical cell? So between those kinds of assays, that's how we're trying to address the question of what other drugs are out there that we could then look at for Angelman syndrome. So I just want to say, as Dr. Silverman said, this really takes a village. Our laboratories work very closely together. Our three labs meet every week. We have a meeting on Angelman syndrome where we coordinate what are the activities that are going on at UC Davis. When is this next batch of rats going to be giving pups? What are the drugs that are lined up for them? What are the therapies that are lined up for them? Oh, we need a lot more mice. We need a lot more rats. That's usually what we talk about. But all of these people are working together every day trying to bring these drugs and therapies closer to being reality. Thank you very much. If anyone has any questions, we'll be happy to address them. There's one out there.
Kristen, do you want to go right there?
Actually, you'll be happy to address them.
Thank you very much for the information you provided. My son is 30 years old, just as a background with a mutation. So my question is, how do you measure speech? In which part? And there's not many animals that have speech like us, and my son has no speech.
So I think one of the really interesting things, and it was a fast-funded initiative, was to create these larger animal models of Angelman syndrome. So of course we can't go into the vivarium where the mouse lives or the rat lives and say, how are you doing today, and gauge how they're functioning. But Liz Berg, or Dr. Liz Berg here in Joel Silverman's lab, has actually done a lot of stuff using the rat model, in that they actually emit high-pitched vocalizations to communicate either excitement, fear, they play. And what they've been able to find is, and it's probably not a one-to-one correlation with what the rat's doing and what the human patient is doing, but at least in these higher order species, these rats have some types of things that show symptomology in terms of how they interact with other animals, how they interact with wild types, that would replicate kind of what we see with Angelman syndrome. So it's not speech, it's not language development, but they do have these different types of ultrasonic vocalizations, of course, that we can't hear, but very sophisticated pieces of equipment can measure, and they can measure change over time. And really the goal is that we can change, or that we can see changes following treatment. And so Liz, who stood up over here, I think would be happy to tell you about how she interacts with these rats, the types of studies that they do with these rats, and truly that they're translational type readouts that we believe will really impact the therapeutic development and understanding of Angelman syndrome.
Hi, for the approach trying to activate the paternal copy of UB3A, what would be the consequence of inadvertently activating things that are next to it? Like, what is SNORD 115, 116, and IPW? And what would be some of the consequences of activating those as a side effect?
You want to?
So we understand a lot of these different SNO RNAs within kind of this chromosome 15 region and their implication in other types of neurodevelopmental disorders, as well as other disorders outside of the central nervous system. I think with a lot of the newer therapeutic approaches are really trying to target some of the spaces between SNORD 115 and UBE3A. So to really kind of avoid any type of dysregulation of these SNO RNAs and other types of regions. There has been a lot of work. We've published work. Dave and Barbara, who's holding the microphone next to you, has pioneered a lot of work where you can disrupt the antisense in totality. And what we do see is that sometimes these SNO RNAs, 116 and 115, can be altered. But again, kind of within the same idea of these different critical windows, their function outside of development is not well understood. So if we're thinking about reactivation of UBE3A in an adult brain or an older developed brain, the impact of these SNO RNAs may not be as impactful than, say, during neurodevelopment. And Dr. Siegel, again, is one of the experts in this area, so if he wants to add to that answer, please.
No, I think he covered it pretty well. But we're definitely trying to keep an eye on all of that. Unfortunately, the mouse models of that would have those regions deleted are not as informative as we would like them to be as to whether that's going to be predictive of problems in people. So if we could have used that as an assay, we definitely would, but that mouse model is not so informative for that. But we want to keep an eye on that with every other model that we work with. And you can be sure that nothing goes into any person until it's proven to be safe.
Okay. Dr. Siegel, if I heard you right, and correct me, You said that Dr. Philpott's lab looked at 2,500 medicines, and one was able to turn on the UBE3A gene.
Yes.
Did I hear you right?
Yes.
Whatever happened to that one that worked?
Yeah, unfortunately, it had very unfavorable characteristics as a drug to treat Angelman syndrome. And I don't say this as any discredit to Dr. Philpott. He's a genius in this field, and that was a very notable achievement. And if anyone would be able to make that drug come to clinical trial, it would be his group. So I know that they're still working on that. They're trying to get around the problems, but unfortunately, it was an easy drug to use in cell culture, but not a very easy drug to use medicinally.
Okay, why don't we take one more question, and then we'll reserve more so we can stay on time for the panel discussion at the end of the day. They'll all be here to answer all of your questions, so write them down.
Thank you. I'm wondering if all the mouse models are the same genotype.
So the mice that are typically used in the Silverman lab that you saw all the wonderful data on, and the EEG and the behavior, those are one of the large deletions of UBE3A. There's, I think, 7 to 10 other types of transgenic animal models of UBE3A that all show different subtle phenotypes depending on what type of, without getting into too much technical detail. And again, the Silverman lab are really brilliant young scientists that would love to tell you about this. But even the genetic background that the mouse is, right? So there's many different types of laboratory mice that we can use. Some of them are more susceptible to seizures. Some of them are very seizure resistant. Some of them show different types of deficits, just depending on how that mouse was derived for laboratory use. And so really what they've done is they've kind of honed in, and why this infrastructure grant that was funded by FAST is so critical is that this is a reproducible, strong phenotype with large effect sizes, meaning that the differences between the wild types and the mice that have this deletion in UBE3A that develop Angelman syndrome, they can do time after time after time. Their deficits in motor, their deficits in cognition remain really, really large, and those large effect sizes allow them for treatment paradigms to come in and be able to measure whether or not a treatment may be more efficacious or less efficacious than another treatment. So they're really focused on a singular genotype for these functional studies, but there are several other that exist in other laboratories across the world.
Thank you. Thank you both very much.